Most of the ocean follows rules that scientists understand. When researchers collect an animal, they usually know where it belongs in the tree of life. A family, a genus, sometimes even the exact species it is. But in the deepest trenches on Earth, that system sometimes fails. Scientists have recovered creatures from nearly 11,000 meters below the surface and compared them with every known species, only to discover that some of them do not fit anywhere. Not misidentified, not rare variants, but organisms with no place in the existing map of life. And that is only one of the surprises hidden at the bottom of the Mariana Trench. When the first cameras finally reached the trench floor, they recorded something scientists did not expect, an ecosystem far more active than biological models had predicted. And inside animals collected from these depths, researchers found microscopic fibers of synthetic plastic, fragments of a surface world that should be impossibly distant. On the seafloor, researchers have observed strange structures scattered across the sediment. At first glance, they resemble small colonies, but closer analysis has revealed something far stranger about what they truly are. So what exactly is living in the deepest ecosystem on Earth? I'm James Carter, and you're watching True Earth. In this video, we descend into the Mariana Trench to explore a world where the rules of biology begin to break down, meet creatures that still have no name, and discover how much we have actually managed to understand about this place after decades of exploration. The physical parameters: 10,924 meters, plus or minus 15, approximately 35,840 feet. That is the confirmed depth of Challenger Deep, the deepest point on Earth. It was measured by multi-beam sonar during the 5 Deeps expedition and published in the Geoscience Data Journal. At that depth, the pressure reaches 1,071 atmospheres. That is roughly 1 metric ton of force pressing on every square centimeter of anything that exists at the bottom of the Mariana Trench. The temperature hovers between 1 and 4 degrees Celsius, just above freezing. And the light? The photic zone, where sunlight penetrates the ocean, ends at roughly 200 meters. The remaining more than 10,700 meters of the trench lie in absolute, permanent darkness. And the floor beneath it is not still. The old, cold Pacific Plate is being forced beneath the younger Mariana Plate at a rate of 2 to 3 centimeters per year, a slow but relentless collision that continues to shape the trench today. Depth drives the pressure, and pressure reshapes the chemistry. Under these conditions, the molecules life depends on should begin to fail. Nothing should survive here. Beat 1.2. Why biology should fail here. Proteins begin to destabilize at pressures between 300 and 800 atmospheres. Enzymes lose function, and cell membranes start to break down. Work later formalized by Paul Yancey established these pressure thresholds for biological stability. Yet Challenger Deep sits at over 1,000 atmospheres, far beyond that limit. Pressure is only part of the problem. Photosynthesis, which powers most ecosystems on Earth, doesn't reach the Hadal zone. There is no sunlight, no plants, no algae. The only organic material reaching Challenger Deep is marine snow, a slow rain of dead matter sinking from the surface ocean. By the time it arrives at Hadal depth, it is degraded, dilute, and scarce. Measurements in the Mariana system show the total mass flux here is among the lowest anywhere on Earth. Before 1960, the scientific consensus was clear. The Hadal zone was a biological desert, a conclusion built into predictive models of ocean life distribution. The term itself was coined in 1956 by Danish marine biologist Anton Brun, who named it after Hades, the Greek underworld. During the Galathea expedition, Brun's team recovered sea cucumbers, polychaete worms, and crustaceans from the Philippine Trench at roughly 10,190 meters, but the dominant model remained unchanged. Beat. 1.3. The first crack, Trieste, 1960. But on January 23, 1960, something did. The bathyscaphe Trieste descended to the floor of Challenger Deep, carrying Jacques Piccard and Don Walsh in the first crewed dive to the deepest point on Earth, approximately 10,911 meters. During the descent, the outer viewport cracked under the immense pressure, and the mission continued. At the bottom, Walsh reported seeing a flatfish-like organism moving across the sediment floor, something alive at a depth where life was not supposed to function. The scientific community challenged the sighting almost immediately, and Walsh later acknowledged the organism may have been a holothurian, a sea cucumber. In 1960, the scientific framework for understanding life under Hadal pressure simply did not yet exist. Beat. 1.3 BY, the model level failure, 2014 JAMSTAC. So Walsh's sighting was largely dismissed as a curiosity, an unresolved footnote. For more than 50 years, the Hadal zone remained at the edge of scientific reach, with only fragmentary observations and disputed sightings informing models that had never been tested at full depth. Then, in 2014, a JAMSTEC deep-sea lander deployed to Challenger Deep during cruise KR-1401 returned footage that changed the picture entirely. Organisms were moving across the sediment floor in complete darkness, under pressures existing biological models considered incompatible with active life at that scale. Subsequent deployments returned the same result. This was not a single anomaly, but clear evidence that the existing models of Hadal biology could not explain what the cameras were recording. The models did not need revision, they needed replacement. The ecosystem that shouldn't exist. Beat 2.1. Marine snow, the energy account that doesn't close. To understand what those cameras were recording, scientists had to start with a basic question. Where does the energy come from? Marine snow does supply organic material to these depths, but the biomass and species diversity documented by lander footage exceed what that limited food source should support. In other words, the cameras were showing more life than the math allowed. Tsati 2.2 Chemosynthesis, the discovery that broke the definition. The answer came from another part of the ocean in 1977, when the submersible Alvin descended to roughly 2,500 meters along the Galapagos Rift. There, scientists discovered dense communities of animals living around hydrothermal vents in complete darkness. Later research revealed the mechanism. Bacteria at these vents oxidize hydrogen sulfide, converting chemical energy directly into organic matter. 4. This was not a minor extension of existing theory. It was the first proof that a complete ecosystem could operate with no sunlight at all. It did not expand the limits of life, it broke them. And the mechanism exists in the Mariana system as well. Cold seep systems discovered in the southern Mariana forearc show the same chemistry that can support chemosynthetic life. In other words, the alternative energy source has already been confirmed near the deepest place on Earth. Beato 2.3. Early discovery moment, the unnamed foundation. With that energy source established, the base of the Hadal food web begins to emerge. When JAMSTEC analyzed sediment cores from Challenger Deep, they expected to find only sparse microbial life, consistent with the limited energy supply. But what they found was microbial density far higher than the models had predicted, and when the genetic sequences were analyzed, a significant portion did not match anything in existing databases, representing entirely new lineages. These microbes are not displaced survivors drifting down from shallower waters. They are endemic and specialized, forming the unseen foundation of the Hadal food web. It begins here, in the sediment, at the base of the entire food web above it. Beat 2.4. Amphipods. Scavengers at the extreme. Above that microbial base, the first visible animals appear, and they are larger than expected. The amphipod Hirundellea gigas dominates the scavenger community at Challenger Deep, with lander deployments documenting dense populations, functioning as the primary recyclers of the Hadal food web. Individuals reach up to 34 millimeters, noticeably larger than their shallow water relatives. Several explanations for deep-sea gigantism have been proposed, including greater oxygen availability in cold water, reduced predation pressure, and the effects of low temperature on metabolic rate, but no single mechanism has been confirmed. Documenting these organisms was never routine. Jameson's freefall lander descended under its own weight and relied on an acoustic release to return to the surface. If that signal failed, the lander and every biological sample it carried were lost. Every specimen recovered represented a risk that could never be fully controlled. On the sediment floor below, other organisms solved the same energy problem in very different ways. Beat 2.5. Sea cucumbers and isopods. Efficiency at the limit. Sea cucumbers dominate the visible life on the floor of Challenger Deep. At some Hadal sites, they make up more than 90% of megafauna biomass, continuously processing sediment for traces of organic matter. Much of what we know about this seafloor comes from footage captured by Jamstek's remotely operated vehicle, Kaiko. On May 29, 2003, Kaiko's secondary cable snapped during surface operations, and the vehicle was lost permanently. The ROV that captured some of the most detailed footage of Challenger Deep no longer exists. Its loss illustrates the cost and risk of deep-sea exploration, and helps explain why so much of this ecosystem remained unseen for years afterward. Beat 2.6. Mariana snailfish plus T. M. AO, the biological climax. But the apex of the Hadal food web is not an invertebrate. It is a fish, and it should not exist at this depth. The Mariana snailfish, Pseudoliparis swirae, was formally described in 2017 after specimens were collected between roughly 6,900 and nearly 8,000 meters, making it the deepest confirmed benthic vertebrate ever recorded. It measures 20 to 28 centimeters, scaleless, translucent, and structurally fragile, a fish that looks like it belongs in shallow water. And yet, this delicate fish keeps pushing the depth record deeper. Observations over the past decade have documented snailfish beyond 8,000 meters, culminating in a record of 8,336 meters in the Izu-Ogasawara Trench, the deepest fish ever recorded. And a depth approaching the biochemical limits of vertebrate life. The mechanism behind this survival has a name: trimethylamine N-oxide, or TMAO. This molecule stabilizes proteins under extreme pressure, preventing them from collapsing at Hadal depths. Measurements show its concentration increases steadily with depth, rising from roughly 40 millimoles per kilogram near the surface to more than 260 millimoles per kilogram in the deep ocean. But that increase has a limit. At extremely high concentrations, TMAO begins to destabilize cell membranes, placing the biochemical ceiling for teleost fish between roughly 8,200 and 8,400 meters. No fish have ever been recorded deeper than 8,336 meters. The most plausible explanation is biochemical. The deepest fish on Earth may already be operating near the limits of what vertebrate biology can sustain. These limits were originally derived from surface conditions, from the size constraints of cells to the stability of proteins under pressure. The Hadal zone does not break these rules. It reveals that they were incomplete. Beat. The 2.7. Xenophyophores, the biological reversal. The snailfish represents the upper limit of vertebrate life in the Hadal zone. But the largest organism in this ecosystem is not its top predator. It is something far stranger, and its existence turns the hierarchy upside down. Xenophyophores are giant single-celled organisms, a form of foraminifera living across the deepest parts of the Mariana Trench. Specimens documented in Sirena Deep reach up to 20 centimeters in diameter, making them the largest individual cells known to science and far beyond the size once thought possible for unicellular life. By aggregating sediment, Xenophyophores create microhabitats that provide structure and refuge for smaller organisms, quietly shaping the seafloor ecosystem around them. This is not a wasteland, but an optimized system, shaped by conditions that should have made life impossible. The ecosystem did not survive the environment, it was built by it. What the data reveals is not struggle, but architecture. CT. Never before seen. Act opening for you. Frontier reframe. For decades, the Mariana Trench was thought to be understood in outline. Its depth had been measured, its pressure calculated, and its biology broadly assumed. In reality, only the outline was known. What lived within it remained largely invisible, because the technology needed to document life at full Hadal depth simply did not exist until 2018. Byte 3.1. The technology that changed the question. Before that year, Hadal research relied mostly on net trawling and early landers without real-time imaging, with no crewed vehicle capable of repeated dives to full ocean depth. That changed with the 5 Deeps expedition, when Victor Vescovo piloted the DSV limiting factor, built by Triton submarines. It was the first submersible designed and certified for repeated dives to full ocean depth across all 5 ocean basins, an operational platform capable of systematic exploration rather than a single historic descent. At the same time, new generations of free-fall lander systems were being deployed by research groups around the world, dramatically expanding direct observation at Hadal depth. Between 2018 and 2024, the volume of high-resolution footage from the Mariana Trench exceeded everything recorded in all previous decades of Hadal research combined. For the first time, scientists were not simply sending instruments into the dark and waiting for what returned. They were descending themselves and staying long enough to observe life directly. The question was no longer whether life exists here, but what life exists here that science has never documented. Byte 3.2. Encounter number 1. The Unclassified amphipods. The 5 Deeps expedition reached Challenger Deep in 2019 and recovered amphipod specimens that did not match any described species in the records. They were sent for taxonomic study, but as of 2023, no formal descriptions had yet been published, and classification remains ongoing. These were not the amphipods introduced earlier. Hirundellea gigas, the known and classified Hadal species, was already documented. The 5 Deep specimens represent additional, genetically uncharacterized lineages living in the same depth zone. The specimens arrived on deck in collection jars and were compared against every documented Hadal amphipod in the taxonomic record. There was no match, not simply new to the location, but new to science. Beat 3.3. Encounter number 2. The unidentified water column forms, and the discoveries did not stop at the seafloor. ROV surveys by NOAA above the Mariana Trench revealed drifting life throughout the midwater column. Among them were organisms the science team could not match to any known classification. Because no AI streams these dives live, the moment unfolded in real time before a public audience. The ROV followed one of the creatures for several minutes, clearly structured and moving, yet absent from every accessible database. It was given an informal field name during the dive and still has no formal scientific name today. Beat 3.4. Encounter number 3, the glass sponge fields. In 2023, expeditions by the Schmidt Ocean Institute documented glass sponge structures deeper than the family had ever been recorded before. The ROV passed the depth where these organisms were thought to disappear, yet the structures continued below it. By existing biological models, they should not have been there. Beat 3.5. The contamination discovery. Another discovery has forced scientists to reconsider every biological sample recovered from this place. In 2019, a study led by Alan Jameson found microplastic fibers in 100% of amphipods, collected from 6 of the deepest ocean trenches, including Challenger Deep at 10,890 meters. The fibers were consistent with synthetic materials from industrial textiles and packaging. The implication is significant. Chemical traces of human industrial materials are now present in the tissues of Hadal organisms, meaning that metabolic and biochemical studies must account for contamination that should not exist in the deepest ecosystems on Earth. Science is discovering new life here, even as it finds traces of the human world within it. Organisms documented for the first time already carrying evidence of a civilization they have never encountered. Beat 3.6. The scale of what remains and the scale of what remains undocumented is staggering. Less than 0.1% of the Hadal zone has ever been directly observed, and only about 400 species from these depths have been formally named. The rest remains unseen, unmapped, unsampled, and unnamed. Ending. Beat 1. Answer the central question. The central question raised at the beginning of this journey has an answer. This ecosystem is powered by chemosynthesis, microbial recycling, and extreme biochemical adaptation, a system that requires no sunlight, no surface warmth, and no connection to the world above. It runs on chemistry and pressure in permanent darkness. It has been operating for roughly 180 million years, while science has known about it for only 60. Ending. Beat 2. The Planetary Echo. In 2024, NASA launched the Europa Clipper mission to study the habitability of Europa's subsurface ocean beneath the ice of Jupiter's moon. Meanwhile, data from the Cassini mission has already revealed molecular hydrogen and silica particles in the plumes of Enceladus, strong evidence of hydrothermal activity beneath its icy crust. If a complete ecosystem can run on chemistry and pressure in permanent darkness on Earth, then the possibility of life elsewhere under similar conditions is no longer philosophical. It is a search parameter. Ending. Beat 3. Final statement. Less than 0.1% of the Hadal zone has ever been observed, and hundreds of recovered specimens still await formal description. Only now has full-depth access become technically repeatable for the first time in history. The question is no longer whether life exists in the deepest places on Earth. For millions of years, the trench has existed in silence, and only now are we beginning to see it.